Hello everyone, my name is Ashfaq Ahmed and I work in the field of bioinformatics and structural biology. Today I will discuss you DFT, Density Functional Theory, also called DFT. I'm making this video on the request of few subscribers of this channel and I hope you will find it helpful. So let's start. What is DFT, how we can perform different analysis, how to interpret the results, and much more. So, in today's video, we will cover basics of DFT, tutorial of performing basic DFT analysis, and interpretation of the results. So, let's start. First of all, open your browser. I'm using Google Chrome. I don't know about you, so you have just to open the browser, write this link colab.research.google.com github/h100. Then click here. You will see this kind of window. It will say H100 AIME. Click here and select this one, Density Functional Theory. Now click the first one, basic of DFT calculation. Click here. Okay, this is the notebook I designed for you and it contains all the informations for learning and for performing DFT analysis. So we will learn and we will perform sideways. So first of all, you need to click it here. Let me connect. I'm going to change the run type. You may not need to change it. You can directly connect to the T4. Otherwise, it's okay. This changing run type is not a necessary step. Okay, click connect. This is necessary. Once the notebook is connected, then we will use some programs uh, for the analysis and for the understanding. These programs include Python, SCF, ASE, Matplotlib, NumPy, IPWidgets, PubChem, Py, RDKit, and ReportLab. The ReportLab will generate a report in the PDF. The RDKit will study the structure of the molecules. From the PubChem Py, we will import the molecules from the PubChem database. From the IP widgets, you will see later on. And the NumPy libraries, and we will use the NumPy libraries, and we will use Matplotlib for plotting. And these are the basic programs will be used for the DFT analysis. So you don't need to read it, but just click here and it will install the necessary libraries. It will take almost a minute, so let's wait. Okay, now it is ready. You can click here and click here, clear selected output. That is optional. Okay. Uh, so we are going to start it formally. We have installed all the programs, each and everything. So you know my name, where I'm working, and I have designed this notebook. I didn't copy from somewhere else. So far, I did not publish this pipeline, but it's okay. You can use it. You can use it for your research and teaching purposes. You are allowed, but if you are using for commercial purposes, you have to contact me. It is prohibited, like directly using it for commercial purposes. Okay, so what is density functional theory? Let me tell you that in this session, I will also teach you the basics, somehow the basics, I will say, and we will also perform sidewise. Okay, so actually the density functional theory analysis or DFT analysis is basically a computational quantum modeling or computational quantum mechanical method. Why we use it? We use it to investigate, to research the electronic structure of many body systems. What do you mean from what does it mean many body systems? For example, a compound is made of many atoms, many molecules. You say not molecules, specifically atoms. So uh, we call it many body system, and DFT is among kind of most versatile method, and it's the most one of the reputable method in computational chemistry and computational physics. But today I will use it like how you can analyze or work with DFT for the different compounds. Okay, so before going to start, you should know the basic concept of these three things. One is Hinberg-Kohn theorem. What is the Hinberg-Kohn theorem? It has two main points or postulates. The first one is that the ground state of many electrons are uniquely determined by the electron density. The second one is the electron density that minimizes the total energy is the true state electron density. Okay, what it means in simple terms, these two postulates means that the state of the atom or the state of the compound, like in which state it is excited state or ground state, or somewhere in the middle, whatever you call it, intermediate state. Okay, so the state of the compound will be elucidated or decided by the electron density. But the true electron density, if you want to measure the true electron density, will be regarded that one which is achieved from the ground state, or in bioinformatics term we call it minimized state. Okay, like just like minima structure minimization, it's the same thing. Okay, but in computational chemistry or chemistry terms we call it the ground state. Okay, now let's discuss the Kohn-Sham equation. The Kohn-Sham equations are set up some consistent like set up some self-consistent equations which allow us to find the electron density. So here, from here we learn that the electron density, the calculation of electron density is the main thing, but which electron density will be regarded true one? The one which is captured from the ground state. But how we will capture the calculation of the electron density? For that, we will use Kohn-Sham equations. These are the different set of equations which calculates this electron density of the system. Okay. Now, the third one is the exchange correlation function. The exchange correlation function accounts for the non-classical part of the electron-electron interaction. Okay. So, now let me summarize that why we are using DFT. This is the main thing you should know, because this was possibly you didn't get me well in these terms or in these explanations. Actually, we check DFT or we perform DFT for many different aspects. The aspects we are interested today is to find out which atom of the compound is more likely to interact. Which atom of the compound is more likely to interact with another in the system. For example, your compound is placed with a protein, I will say like this, and then which one is more reactive atom of that compound. Let's say your compound name is water. Let's say your compound name is benzene. Okay, so which atom is more reactive? So how we will find it? We will find those things from the DFT analysis. And what we do in the DFT analysis? In the DFT analysis, we actually calculate the electron density. Which electron density will be the true one? The one which captures from the ground state. How we will calculate the electron density? By Kohn-Sham equations. Okay. So now this was just the brief conclusion of this whole thing. Now we are going to set up a simple DFT calculation. Remember, in this setup DFT, in this simple DFT calculations, we this is the code you will not see initially, but if you want to see, just click the show code. You will see now. So here we just we import some libraries, the DFT library and the GTO library from the PyCSF, and then we define a molecule, like which molecule we are calculating the DFT. So we are calculating the DFT on H2O, or we say it water, H2O. So here we place the H2O molecule, and then we are using a model STO3G, and we will learn it about these models later on. And then we perform some the DFT calculations, and then we print the total energy. Okay, so let's click here. Now you can see the total energy or the converge is CF, both are the same thing almost. So they are, this is the total energy of your compound. It means the compound is possibly in ground state. Remember, uh, there are two terms in physics, I would say, enthalpy and entropy. Both these two terms are related to each other. Entropy explains the orderness or the disorderness of the system, and the entropy will be higher if the delta H or enthalpy or the change in energy is higher. So in that aspect, everything in the ground state, we call it native state, even if it is proteins, and that is why we are always minimizing structures during docking and other things, because we want to ensure that the structure we are using is in minimized condition, or in the ground state or the native state, whatever you call it. Okay, so here I cleared that question also. Okay, so now I want to visualize these calculations performed. These are just simple calculation I performed, just calculated the energies, and now I want to visualize. Okay, so how you will visualize? Just click here. It may take some time. Now you can see here, this is the HOMO of water molecule. What is HOMO? We will learn below. Okay, so this is just the HOMO of water molecule, because we have calculated DFT for water molecule here. So this is the HOMO. At this stage, you need to just understand the structure. This is the legends. This is the blue is the positive, and the red redness shows the negative value, and the colors actually represents or indicate the electron density. And you can say these two dots are basically the electron, and this is the density. Okay, now we will explain it here. Okay, now here it is, it says that calculate for the molecule of your choice. Okay, so we will just skip it for this time. Okay, now we are coming to the interpretation of this thing, because the interpretation is necessary for understanding of the DFT, or to work in the DFT you have some solid or some initial understandings. Okay, so how to interpret these results initially, then we will run, or we will understand how we can use our own molecules for these kinds of analysis. So for the understanding is also so much theoretical, but I will try to make it, to present it on the easiest way. Okay, HOMO stands for highest occupied molecular orbital. Okay, here before going to explain or to understand these things, you need to understand, or I want to remind you few basic concepts from the chemistry. Okay, there is a word named orbital or orbital. So in orbital, for every atom, there is an electron density. It means the electron density means that the electrons are in constant motion around the center of that atom. It's just like this, okay? The simple way, okay? Now you assume it like there is some object that is a center of the electron atom, and the electrons are in constant motion in different paths, just like galaxy. Okay. Now these orbitals are actually those paths of the electron they are moving in. For example, the electrons are moving in some path, so around that center, so that is actually the orbital. Now remember that some of the electrons are very naughty. I will use the word naughty, okay? Or they are more reactive compared to the other electrons. For example, if an atom has 8 electrons, 2 in the down shell and then 6 in the other shell, and then I don't know, 6 or 8. Okay, let's say 6 in the other shell and then 8 in the other shell. So in that case, the topmost orbitals, which are exposed to the outside, from which are far away from the center of the atom, they are normally interacting. They are normally taking part in different bonds. They are normally forming different bonds, different interactions, like hydrogen bond, ionic interactions, and blah blah blah. So our main target, or the concept of DFT basically targets the electrons which are present in the upper orbitals and the higher orbitals. So, and from there we will assess that which atom of the molecule has a tendency, more vibrations or more thermodynamics for interactions. Okay, so now you understand what the HOMO means, highest occupational orbital. So let's come to this figure. Oxygen has two lone pairs of electrons. Two lone pairs of electrons. It means that the oxygen has two electrons in the outside shell. So these are actually those two electrons of water, because we have used water and the water contain oxygen. Okay, so highest occupied molecular orbital, it actually visualizes the electron density distribution in which orbital, the highest energy orbital, and the highest energy orbitals are normally the one which are far away from the center of the atom. Okay, the orbital is significant because it plays a role in chemical reactions, particularly in the process like electron donation, or to what we call redox reactions. Redox reaction means reduction and oxidation. and bond formations. I explained this point already to you. Okay, what does the HOMO represent? So I already explained this also, but it will be easy for you now to understand. The HOMO represent the most loosely bounded electrons in the molecules. Now you got my point. For example, we have benzene structure. Okay, so benzene is a ring structure which has six carbon. Okay, six carbon ring. So how many atoms are there? So many, six are carbon and some hydrogen and blah blah. Okay, you can assume some any other other molecule. Assume so that molecule which you have assumed, it contains different atoms, right? If we assume water contains three atoms, H, two hydrogen and one oxygen. So the same way you can assume any compound, and that compound contains different atoms. So what the DFT is looking for, the DFT or the HOMO represent the most loosely bounded electron. If this compound contains some atoms which has some loosely bound electrons, I will call it naughty electrons. So technically those loosely bound electrons, or I would say out of control electrons, okay, just for this fun, just for fun. So those out of control electrons will be most likely to involve in interaction. So we will say that yes, in this atom, in this compound, this atom has the tendency to form the interaction as compared to the other atoms present in this compound. I hope you understand my point. These electrons are most likely to participate in chemical reaction. Why? Because they are loosely bound electron. Why? Because they are present on the higher energy orbital. Why? Because they are far away from the center. They are out of control, say this way, for a conceptual logic. Okay? So such as forming bonds and other molecules. So the shape of the distribution of HOMO provides insights into the reactivity and electronic properties of molecule. So this kind of figure, when you receive for your compound, so it will actually tells you the shape, like what kind of result you are expecting, what kind of results you are going to expect, which atom is more reactive or which atom is less reactive. So these things actually gives you the electronic properties. Okay, now if you are new in the field, so a question may come in your mind, so what I look for in this figure. So key features to look for in the HOMO figure. The first thing, electron density. The brighter region, often color red or blue in the plot, represent the area of high electron density. So these brighter regions, these, whether they are red or whether they are blue, they represent high electron density regions. Okay. The dim regions, often white or transparent, they represent low or no electron density. It means these regions. Okay. The electron density is typically concentrated around atom or bonds where the electrons are most likely to be found. Okay, it's easy. Okay. The second thing, you look for the symmetry and shape. The HOMO often reflects the symmetry of the molecule. For example, in a symmetric molecule like benzene, the HOMO will have the symmetric distribution of electron density. So in some molecule are symmetric or some are asymmetric. So in asymmetric or in symmetric, like benzene, the the distribution will be like that. In this case, we use the H2O, and this is the electron, the two lone pair, the density of the two lone pair. So that's why a benzene, so it's not a symmetric. Okay. The shape of the HOMO can be bonding, anti-bonding, or non-bonding. So these are also technical terms you need to understand, that whether this shape of your compound, it comes in bonding orbital or anti-bonding or non-bonding. How will you understand? Electron density is concentrated between atoms, so it will indicate stabilizing interaction. For your compound, if the electron density is concentrated between atoms, we will call it stabilizing interaction. If the electron density is concentrated outside the bond region, we will say destabilizing interaction. For example, you know that this is the bond region. Let's say this is not the bond region in this case, but let's say this is the bond region. Let's say this is benzene, so this is carbon and this is carbon, and this is the bonding region. This carbon and this carbon, take it this way. And if the electron density is concentrated outside the bond region, it's not concentrated on the bond region, then we will say it's a kind of destabilizing interaction. And the third one, non-bonding orbitals. If the electron density is localized on a single atom, we also call it lone pairs, then we will call it non-bonding orbitals. It's just like the same. For example, you have two atoms, but one of them is flashy, one of them is not flashy. It means one of them has a light or the electron density or naughty electrons, and the other do not have or does not have. So that's why it will be kind of non-bonding orbital. Okay. The third thing you should look for, the localization versus delocalization. If the electron density is localized on a simple atom or bond, it suggests that the HOMO is associated with a particular functional group of the region of the molecule. If the electron density is delocalized, it means that the electrons are shared across large regions of the molecule. Okay, just like in case of these things. Okay, I want to tell you one thing, just for a break of 30 seconds. Basically, I'm not a chemist. Okay, so if you are a chemist and you think I'm interpreting something wrongly, so just pardon me, because basically I'm not a chemist, and I accept those smaller misinterpretation, possibly because of language barrier or possibly because of my limited knowledge. So I excuse already. My my main aim is to just convey how to perform and how to do these things. For expert view, you need to read different research papers and books and etc. for too much expertise. Okay, interpretation of the HOMO for chemical reactivity. So how you will interpret the HOMO graph, like whether the chemical reactions are taking place or not. Regions with high electron density in the HOMO are potential nucleophilic attack. It means they will donate electrons. If the density is higher, what I say, like these are the lone pair of the oxygen, you can see the density is higher. It means in water, when water interact with other uh, molecules in the system, so these two atoms will be ready to donate. Water can donate these atoms and they can generate a bond. Okay, so electron donation, the HOMO is the orbital from which electron are most likely donated in the region, almost same thing. Okay, reactivity trends. Molecules with higher energy HOMO are generally more reactive because their electrons are less tightly bound. Let's say you compare this figure with the HOMO of another molecule, and you see not brighter spot like this in that molecule. So you can say that water is more reactive as compared to that one, because the density higher or the flashy sides are here, more here compared to that other compound. Okay, now specifically we are interpreting. These were the general rules. Now we are going to interpret specifically these things. Okay, so let's say interpret the water. Let's take an example of water, which we use in the DFT calculation earlier. The HOMO of water is primarily localized on the oxygen atom, representing lone pairs of electrons I showed you above. The electron density is concentrated the oxygen with little to no density on the hydrogen atom. So that means that the hydrogen atom did not show any density. The two brighter spots you see are basically from the lone pair of the oxygen. This indicates that the oxygen atom is most likely site for the electron donation during hydrogen bonding or nucleophilic reaction. So now if you have this kind of calculation, then if someone asks you, okay, how can you say that oxygen is taking place in the hydrogen bonding, not the hydrogen? So the answer is because the lone pair, I performed DFT analysis, and the lone pair of the oxygen, they show more electron density and they are in the higher orbital, and they are more likely, they are more reactive than hydrogen. So therefore the oxygen atom is more likely to donate or to take part in the, how to say, in a chemical reaction. I hope that was fair enough. Okay, so now we are coming to 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 understand, like this was just I ready-madely I used water in this analysis. So now I'm trying to use any other molecule. So for the sake of that, I'm going to open PubChem. If you don't know, PubChem is a database and it contains almost any compound. Let's say I'm interested in, let's say, caffeine. Okay, so can you see this is a compound CID 2519. So the test compounds of your choice from the PubChem with preliminary report. Okay, first we run this one. Okay, so what you need to do, just click here. Once you click here, you will see this thing here. Now, what was your compound 2541? Let's say try another one, not caffeine. Let's say I'm trying captopril. Okay, this is 44093. This is a compound. Okay, 44093. 44093. Okay, now what you will do, click here, calculate HOMO, and then wait for some time. It will fetch the molecule from the PubChem. It says that this is the molecular name. Uh, I call it captopril, but the exact name is this one, IUPAC name. You can read here, and the calculation is smart enough. The code is smart enough to pick, and then this is the molecular formula. This is a smileys, but wait for it. It will generate the HOMO figure, and depends on the complexity of the compound. It will takes maybe 30 minutes, 30 seconds or 1 minute. So okay, this is a kind of HOMO figure. Wait, molecular name. Okay, this HOMO figure, at times it will generate a very good HOMO figure, and at times it will generate some bumpy areas. That you can see these are some bumps. But I have dropped two codes for you. So let's try this code. If in your case, if this code does not work well, so then we will try this code. Let's see what it say. 44093. Okay. What is the difference between this one and this one? This code will also generate a PDF here, PDF report here, and also it will generate a report here. But but you have to read this warning message. The below code generates a kind of preliminary test report. However, this code is in testing phase, and I personally did not test it on the different compounds. Therefore, I strongly suggest to please consult an expert in case you want to publish it. What I mean, because to be very real on the realistic side, I generated these codes last night, less than 24 minutes, 24 hours, and I did not have much more time to just go on and test it on different compounds. So I kept it here. So I cannot take the guarantee of the report. So now you can see this here, this code somehow failed, but this code generate a very good interpretation. Also, it will generate another thing. We have to wait it. It will also generate a kind of 3D image or the 3D surface image. We have to wait. You have to wait until you do not see how to say a PDF report. Although it has generated, okay, it has generated the this thing, but the report is in progress, so we will wait. So far we can, okay, it's generated. Now the problem with this figure is that uh, you may not be able to clearly see the electron density here. Although I will try to fix it and will update this code if you are interested in this kind of image. But when I will fix, it's another debate, but I will try. So far, let's download this code. Okay, now you can see a report like the HOMO analysis. This is the total energy of the captopril, maximum electron density, minimum. HOMO is asymmetric localization, HOMO is delocalized. But listen, as I told you that I do not guarantee this report. Possibly it will be correct for some and might be correct for everyone, might be incorrect for all of them, but still I tried myself to the code should generate a report, but I cannot guarantee its correctness. So you should have to consult some expert. Okay. Now these are the two individual images for the ISO surface, and this thing you can download it. You can download, let's say, why not we try benzene and to see the behavior of this code. Benzene is 241, and let's see. Uh, okay, first you have to run this. When you run this, it will ask you for 241. Now click calculate HOMO. Let's see how this code behaves and how the down code behaves. So it will give you, yes, this is somehow great. You can see 1, 2, and then maybe one here, 3, 4, 5, and another one is here. But the electron density, it's clear the electron density is higher on these two atoms in benzene, not on the other. And let's say to just first run this, then type 241 the CID of your molecule. Oh, sorry. Type 241 and then click here, generate HOMO, R and HOMO report, and it will pick name and each and everything. It will give you the energy, it will give you the HOMO analysis. This one is far better. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5, and 1 is here. So once again, those two atoms are um, shown with the high electron density. Okay, so that was about the basic electron density. And here, if you want to explain the benzene figure, so you can use it. And these are some limitation about the HOMO. Please read it. And finally, I will also make another video where we will calculate the vibrational frequency and the thermodynamics properties of some particular atoms of the compound. So, lastly, I just want to share a few things just shortly. Okay, number one, use this code on your own risk because I haven't tested critically or extensively, or they are not published. So you can use it, doesn't matter, not for commercial, but for academic and research. And also, the second thing is that if you are interested in these things, just subscribe this channel and wait for another video so you will not miss it. And the third thing is that I forgot. Okay. Okay, so I hope you like the density functional theory. And remember, I work in the field of structure biology and bioinformatics. I'm not a chemist by nature. So once again, I apologize to the people basically from chemistry if I made a mistake during interpretation. So that's it, and it was a highly requested video uh, from some of the subscribers, so that's why I made it, and I will also make the second part soon. So okay, thank you so much. Bye bye.